Good day everyone, I'm Lovely Bias from BSEd 2D Science. This video will demonstrate to you the different stages of mitosis and meiosis. Animal cell would be the basis to perform this activity. And I have here the different materials which will serve as your legend so as to understand well this activity. This is the nucleus. This is the nuclear membrane, nucleolus, maternal chromosome and the centromere, paternal chromosome and the centromere, spindle fiber, and the centrioles. The cell cycle consists of four distinct phases. First is the G1 phase. It is the time when the cell grows and performs its natural function. So what comes after G1 phase? The S phase or synthesis. It is the time when the DNA is replicated, means every chromosome is in doubled form. The two chromatids formed by the site of centromere. So what comes after the S phase? The G2 phase, or collectively known as the interphase. It is getting ready for the cell division. So what comes after the G2 phase? The M phase or mitosis. Mitosis is a series of changes in the nucleus, including an exact duplication of complete set of chromosomes, and the separation of these chromosomes into two identical sets of chromosomes. Mitosis is subdivided into four stages, which are prophase, metaphase, anaphase, and telophase. The first stage is the prophase. At this stage, the chromatin begins to condense and appear as visible chromosomes because of the two sister chromatids that are pinched together by the site of centromere. The special protein attached to the centromere to form kinetochore. Finally, the nuclear membrane disintegrates. And the nucleolus begins to disappear. The centrioles begin moving to separate to opposite ends of the cell and forms spindle fiber that helps to separate the chromosomes. Next stage is the metaphase. At this stage, the chromosomes align themselves at the mid-region of the cell nucleus. Each chromosome is connected to a spindle fiber at its centromere. This line is referred to as the metaphase plate. Next stage is the anaphase. At this stage, enzymes break down the proteins in the centromeres and then splits, which allow the sister chromatids start to separate and move apart. Once the chromatids separate, they are considered to be individual chromosomes. Next is the telophase. At this stage, when the chromatids reach the opposite poles of cell, a new nucleolus begins to reform around the chromosomes, forming two daughter nuclei. The spindle fibers disintegrate, leaving the centrosomes just outside the nuclear membrane. The nucleolus begins to reappear. Once the nuclear membrane starts to reappear around the nucleus, mitosis is complete. Meiosis 1 is results to two haploid daughter cells, each with half the number of chromosomes as the original cell. Next is meiosis 1. The first stage is prophase 1. Just like in mitosis, the nuclear membrane and nucleoli disappear. The spindle fiber starts to form. But the difference is what is going on with the chromosomes in this prophase. At this stage, each chromosome pairs with its corresponding homologous chromosome to form a tetrad in a process called synapsis. Along with that, crossing over occurs in prophase 1. When we say crossing over, it requires the chromosomes break and reconnect to the other chromosome. Next is the metaphase 1. Tetrads line up at the equator. The spindle fibers attach to the chromosomes. Same with the prophase 1. The genetic recombination is occurring also in this stage, the metaphase 1. Next stage is anaphase 1. As soon as anaphase 1 gets going, genetic recombination is over. The spindle fibers pull the homologous chromosomes toward opposite of the cell. Next is the telophase 1. Chromosomes with two chromatids decondense, and a nuclear membrane reforms around them. Each nucleus is now haploid. Meiosis 2 is occurs after meiosis 1, which covers prophase 1 to telophase 2. It is really similar to mitosis. The only difference is that the two chromatids per chromosome are not necessarily identical due to genetic recombination occurring in meiosis 1. Next is meiosis 2. The first stage is prophase 2. During prophase 2, the chromosomes with two chromatids become visible as they condense. The nuclear membrane and the nucleoli disappear. The spindle fiber is forming. Next stage is metaphase 2. The chromosomes with two chromatids line up in the middle of the spindle fibers along with the equatorial plate. The anaphase 2. The sister chromatids separate or split and move toward opposite pole of the cell. The last stage is the telophase 2. Chromosomes with only one chromatid condense and get surrounded by new nuclear membrane. Now, it results in 4 haploid daughter cells and have the right amount of DNA.